In the last module, we concluded that pyruvate was the final end product of glycolysis. What we're going to do today is expand on the potential fate of pyruvate when oxygen is available, which is known as aerobic metabolism. So just briefly, aerobic metabolism is aptly named aerobic because it requires oxygen for the creation of ATP. Without it, this process doesn't happen at all, and hence the name anaerobic for the methods of glycolysis and phosphocreatine systems, because they don't require oxygen, and aerobic here. So when oxygen is present in sufficient concentrations, pyruvate molecules from glycolysis can then be used for the aerobic production of ATP. This occurs then all in our mitochondria. You'll recall that mitochondria are called the powerhouse of the cell, right? That's because that's where the majority of the ATP is found, and will lead you to believe now that as we get into aerobic metabolism, that this is going to generate much more ATP than the single ATP generated from phosphocreatine system or the net 2 ATP from glycolysis. Aerobic metabolism ultimately involves the interactions of two pathways. One, the Krebs cycle, or otherwise known as the citric acid cycle, or TCA cycle. You'll hear me use the TCA cycle most often. Your book and these notes use the Krebs cycle. Just know they're the exact same thing. Ultimately, the Krebs cycle, the goal is to break down all of the carbons from a glucose molecule, create these high energy molecules in FADH2 and NADH2, and then pass them on to the second cooperating metabolic pathway, which is the electron transport chain, most commonly abbreviated as the ETC. What I'll show you next is a video of how the ETC works, but ultimately what will happen is oxygen is not a direct participant in the Krebs cycle or in the electron transport chain, but what it does is it acts as the final hydrogen acceptor at the end of the electron transport chain, ultimately taking two hydrogens, combining them with that said oxygen to create water. In lab, this relates, of course, to the measurement of oxygen consumption, or VO2, right? Oxygen isn't really consumed, it's just created, it's just transformed in its form from free oxygen then to water, and therefore is called consumed despite not actually being consumed and going away. The aerobic production then has a couple names. We'll call it oxidative phosphorylation. Sorry, again, that's because we require oxygen to phosphorylate ATP at the end of the electron transport chain, or the other very common term you will hear is called cellular respiration. Respiration leading to that idea of the breathing of oxygen in this case, or the usage of oxygen to form ATP. So what I'm going to do next is I'm going to play a video that will really walk you through both the Krebs cycle and the electron transport chain. It'll show the movement of these atoms and ions in a much better way than I can depict in a video. After the video, we'll come back and we'll chat about the important things and clarify a few ideas.
When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain. The electron transport chain is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP. In summary, we have seen how the 4 stages of cellular respiration are responsible for converting the energy found in the glucose molecule into ATP, the energy battery of the cell. On average, 36 ATP molecules are produced per glucose molecule that entered the cell. In the process of producing ATP, oxygen is brought in from the bloodstream to be the final electron acceptor in the electron transport chain, and the carbon dioxide that is produced as a byproduct is released.
I hope you enjoyed that video and being able to see the moving pieces that's involved in both the Krebs cycle and really in that electron transport chain. What we're going to do now to finish out this lesson is walk through each step a little bit with slides here so that you have another visual, have some words on the screen that you can read and help study. If you're still confused, I highly recommend using the book as a great resource, or YouTube is always a fantastic resource for learning and hearing about how this process works from other people and seeing it moving. Okay, so to get started, we can think of this as a 3-stage process. The first stage really isn't part of oxidative metabolism per se, but it's something that has to happen. So if you remember, in glycolysis, the end product is pyruvate. Pyruvate is generated in the sarcoplasm here and transported across the mitochondrial matrix or the membrane, and when it is found in the mitochondria, it can then be converted to acetyl-CoA. In that conversion to acetyl-CoA, we end up losing 1 carbon, 2 oxygen molecules in the form of CO2, which can then leave the body and then be breathed out, exhaled. And we also reduce an NAD into an NADH as shown here. So acetyl-CoA is now a 2-carbon molecule because we lost one of the carbons. It then combines with oxaloacetate and enters the Krebs cycle. So again, acetyl-CoA, oxaloacetate is a 4-carbon molecule, and they form citrate, which is a 6-carbon molecule. There's a series of reactions that follow, known as the Krebs cycle, that ultimately will reproduce oxaloacetate at the end as a 4-carbon molecule. Hence the name Krebs or TCA cycle, because it essentially makes a cycle, right? In this series, what do you need to know? So here's what you need to know. You need to know that it begins and ends with oxaloacetate. We need to know the products that come out of this. So we need to know that 3 NADHs by stars and an FADH are produced. We have a substrate level phosphorylation with the creation of GTP, which we can then convert to an ATP to function just like a normal ATP, and we produce 2 molecules of CO2. And so you say, okay, why are all these steps in here? What I need you to kind of think about is not necessarily knowing the names of the steps, but understand that as each of these steps takes place, the idea that we can promote an increase in the activity of some of these enzymes to help speed these up and slow them down depending on very key steps. So just know that the products that come from this, which we'll talk about here in a little bit, and then just know that these steps are also alterable. We can speed up or slow down these enzymes to increase the rate of the Krebs cycle. I'll make one other final point at this point. So we produce 2 CO2 molecules from the Krebs cycle and 1 CO2 from the creation of acetyl-CoA. So that leaves us with 3 CO2s produced. And you may ask, where do these 3 CO2s come from and what is the importance? Ultimately, oxidative metabolism is taking glucose, which is a 6-carbon molecule, and using the energy bonds found in each carbon molecule to then produce FADH and NADH. Those NADH and FADHs are then used to create ATP. So ultimately, we're going to have to cleave the bond of every single carbon dioxide in there. So you'll remember glucose in glycolysis goes from a 6-carbon to a 3-carbon molecule. That's here. So pyruvate is the ultimate result of that 3-carbon molecule. If we pass this through twice, we get 6 carbons. So as you can see, we fully break the bonds of all 6 carbons in a glucose molecule to create these reduced reagents that we can then pass on to the electron transport chain. If we specifically look at the Krebs cycle tally so far, here's what's happened specific in the Krebs cycle. Remember, glucose to pyruvate, 2 acetyl-CoAs, then feed into the system. Will generate 3 NADHs, an FADH, 1 GTP, which then ultimately is substrate phosphorylation and creates an ATP, and 2 carbon dioxide molecules. And again, since we split glucose in half, we get 2 of these. So total from the Krebs cycle only, we get 6 NADHs, 2 FADHs, 2 ATP ultimately, and 4 CO2. We can also add this to the acetyl-CoA step. So don't forget these, but in this we'll have 2 CO2s and 2 NADHs. So note that oxygen has not been utilized, right? As you saw in the video, oxygen has no part in the Krebs cycle. It's only at the very, very end of the electron transport chain. So that brings us to ultimately oxidative phosphorylation or electron or cellular respiration. This is an important process because the vast majority of ATP formed in cells comes from oxidative metabolism. You can just see we're building this energy and building this energy to ultimately create a large number as opposed to 1 or 2 from anaerobic for each chain. The theory is that the electron transport chain works by pumping hydrogen ions out of the mitochondrial matrix in order to generate a potential energy. So you push them out, you create this barrier of the mitochondrial matrix, and all the hydrogen ions want to come back in, but are unable to because the membrane is not permeable to them. We then couple them by using ATP synthase, which is a windmill. Actually, it looks like a windmill if you look at the structure, to then allow those hydrogens to flow. It spins like a windmill to then create an energy system to couple ADP and drive the phosphorylation of ATP. So the term oxidative phosphorylation does not mean that oxygen necessarily participates in the phosphorylation process. However, because oxygen is such a great acceptor of electrons, it is a strong oxidizing agent, and therefore we can use it at the end of the electron transport chain and allow oxygen to act as the final electron acceptor at the end of the chain of oxidation reaction potentials. Remember, as we pass through, we'll see electrons are being passed down the electron transport chain, and we have to have somewhere for them to go at the end of it. Oxygen is going to be that perfect acceptor. So what does it look like? So we can say that there are 4 complexes in the electron transport chain. Complex 1, complex 2, complex 3, and complex 4. You need to know not the specific names of the complexes, but know that there are 4 complexes, because that's going to come into play. So complex 1 is specific for NADH. NADH actually skips complex 2, which I know is kind of confusing, but it skips complex 2. FADH adds its electrons in at complex 2, and then it goes to 3. So for NADH, we go NADH into 1 to 3 to 4 to ATP synthase. FADH goes from 2 to 3 to 4 to ATP synthase. So just keep that in mind. So again, well, the idea is that we're using electrons to pump hydrogen ions outside against their concentration gradient. And as you can see, the gradient is high here. There are lots and lots of hydrogen ions just wanting to get back, completely impermeable. So as we pass electrons from complex to complex to complex, that allows us to pump protons out to essentially accumulate a source of potential energy. And a good example of this is the idea of like damming up a river, such that as that water then falls over in the world, at least, we can capture that water falling over to actually generate electricity in a dam. Right? The same idea is going on here. We're capturing all this potential energy that we're then going to be able to use to drive our ATP synthase. And again, you can see here, at the end of the day, we have electrons that are passed out of complex 4. Those electrons have to go somewhere. So oxygen acts as the final electron acceptor, binding to 2 hydrogens and becoming water. In lab, we talk about this, and I'll just highlight it very briefly, but we talk about oxygen consumption. And the idea is that oxygen actually isn't being consumed in any reaction. Again, it's that oxygen is becoming water by acting as this final electron acceptor and then allowing this process to continue. If we have no oxygen available, then this electron essentially gets stuck right here, is the idea. So there's an electron right here that's waiting to be accepted. If it doesn't have anywhere to go, it stops. When it stops, this process stops. This process stops, and this process stops. And all aerobic metabolism stops unless oxygen is there to capture it. So how many ATP then do we produce for each run of an NADH or FADH through the electron transport chain? So we can calculate that. So first, through the first pump, which is complex 1, an NADH will pass its electron on and will pump 4 protons. The second pump, which is confusing, is actually complex 3. And again, as we move electrons down that, we will also move 4 protons across. And then in the third pump, which is complex 4, we will pump 2 across. Okay. Some really smart and bright biochemists have determined that it takes 4 hydrogen protons are required to produce 1 ATP molecule when the hydrogen ions come back down through ATP synthase. So if we add them all up, we can say that, if you remember, NADH comes in at complex 1, so therefore we get 4 plus 2 gives us 10 hydrogen ions pumped across per molecule of NADH, giving us a net of 2.5 ATP per NADH. If you'll remember, in the previous slide, I introduced that FADH does not enter complex 1. It actually enters its hydrogen or its electrons into complex 2. As you'll see here, complex 2 is not responsible for pumping any protons across, and so we get no proton motive force or increase in ATP from that step. However, we then do pass that electron onto complex 3 and complex 4, giving us a total of 4 plus 2 gives us 6 hydrogens, and 6 divided by 4 gives us 1.5 ATP per FADH generated in the TCA cycle. So let's get a final tally of ATP for carbohydrate metabolism. So we'll start all the way at the top. Glucose comes in. It is phosphorylated, and we lose 1 ATP in that phosphorylation step, but that's important because it keeps it into the cell. Phosphofructokinase, or PFK, phosphorylates it a second time, losing ATP number 2. However, at the end of the day, we then produce 4 total ATP in that step. We also generate 2 molecules of NADH in glycolysis that we can then ship to the electron transport chain and use that. So here we have 2 molecules, 2.5 ATP per molecule gives us a total of 5 ATP from those molecules, assuming oxidative metabolism. In pyruvate, we have 2 molecules of NADH that are produced that can then be sent to the electron transport chain. Again, 2.5 times 2 gives us a total of 5 ATP produced. As we go through the Krebs cycle, we produce 2 GTPs, which ultimately immediately become ATP and can be thought of as similar as that. We create 3 molecules of NADH per cycle, but remember, 2 cycles through gives us a total of 6. 6 times 2.5 equals 15. And then we get 2 molecules of FADH, 2, and we can multiply that times 1.5 ATP for a total of 3 ATP. If you add them all up and subtract these 2, you get a total of 32 molecules of ATP per glucose. The book also has this for glycogen. I won't walk through it all again, but in glycogen you'll see the only difference is that when we cleave the molecule from glycogen, it is automatically already glucose 6-phosphate, and therefore we don't have this extra input in here. Therefore our yield is 32 and 33. So I would definitely be able to, if I were you studying for the test, definitely be able to walk through each of these steps, knowing how these ATP are created, knowing where they go, how many, and then being able to add up all of these steps and generate ATP from them. That ends the carb lecture. We're going to jump into lipids in the next section.